Over the course of this semester, we conducted a series of conservation biology analyses on a set of paired plots on campus. For this independent project, we selected plots 8. Our developed plot, circled in blue, is located in front of the Maryville University Library. The wild plot, circled in red, is located behind McNally House and expands behind the Design and Visual Arts Studio. As you can see, the developed plot is well maintained and very open with few trees. Most of the diversity is a result of the potted plants and decorative flowers around the perimeter of the plot. These plants were changed out seasonally. With the exception of the trails along the edges, the wild plot was much less controlled and allowed a variety of species to grow. There was less human involvement here, so we heard more birds and noticed a significantly higher amount of plant growth at this plot upon every visit.
Our first plant that we were talking about is honeysuckle. It is an invasive plant that was all over our rural plot. We recorded over 40 honeysuckles. Our next plant is lantana. It is a decorative plant that we found in front of the DUC in our urban plot. There were 12 recorded. Our last plant is bald cypress. It was found in our rural plot. We found only one bald cypress, but there were many other conifers found. Our first bird was the northern cardinal. Northern cardinals are present in both rural and urban plots. We found 6 northern cardinals in total. Our next bird is the northern flicker. Northern flickers are found in our rural plot, and they can be found in Missouri year-round. Our last bird is the blue jay. Blue jays were present in both our rural and urban plots. Blue jays can be found in most of the eastern and central part of the United States. The alpha diversity for plants in our urban plot was 18, while our alpha diversity for plants in our rural plot was 22. Our alpha diversity for birds in urban plots was 6, while for birds in rural environments it was 11. Our alpha diversity indicates that there is a moderate level of diversity in urban plants, with rural plants having a higher level of diversity. Urban birds showed a low diversity, likely due to habitat suitability. Rural birds showed a higher alpha diversity, meaning more species are suited to the rural environment. Our gamma diversity for plants was 40, while for birds it was 17. For our gamma diversity, plants had a much higher gamma diversity than birds. This indicates a very high level of species richness. The gamma for birds being lower than that of plants suggests that bird diversity is lower in this region. Our beta diversity is highest in both plants and birds in rural environments. Low beta diversity in urban environments suggests there are limited niches and species turnover. This indicates that urban environments support less specialized species. Our beta diversity for plants in urban environments was 0.45. Our beta diversity for plants in our rural plot was 0.55. For birds, our beta diversity in our urban environment was 0.353, while in our rural environment it was 0.647.
The Shannon Diversity Index will tell us the diversity of a community. We will find it using the equation negative sum of the proportion of total individuals in a species times the natural log of the proportion of total individuals in that same species. Using this equation, we were able to find the Shannon Diversity Index for plants in both the wild and developed plot. Despite having a higher alpha diversity score, the wild plot has a lower Shannon Diversity Index. This is because the species found there were not as evenly distributed as the species found in the developed plot. Some plants, like goldenrod, had as many as 100 individuals, increasing the proportion in relation to the total number of individuals. Both plots, however, indicate a healthy level of diversity as found through these values. Similarly, we were able to calculate the diversity of birds in both plots. The Shannon Diversity Index for birds in the developed plot was lower than that of birds in the wild plot. This indicates that the developed plot is not as species rich or abundant as the wild plot.